Good afternoon, everybody. The class is about salivary secretion. As we discussed in the last class about the functions of GIT, it was an overview view about the whole gastrointestinal tract. As you all know, there are 5 secretions: the salivary, the gastric, the intestinal secretion, the bile, and the pancreatic. Today's class is about salivary secretion. Saliva is secreted. We will deal the objectives of the classes. We should know about the three salivary glands, their anatomy, nerve supply, the composition and character of the saliva, functions, regulation of saliva, and applied aspects. There are 3 glands: the parotid gland, which secretes the serous secretion, the submandibular, which is both serous and mucus secretion, and the sublingual, which is the mucus secretion. The parotid gland opens on the inner surface of the cheek, opposite the second molar tooth, by the duct of Stenson. The submandibular gland opens by the Wharton's duct in the floor of the mouth by the side of the frenulum of the tongue. The sublingual gland opens by several fine ducts in the floor of the mouth on either side of frenulum of the tongue. This is called ducts of Ravenous. So the parotid gland, which has the ducts of Stenson, the ducts of Wharton, the ducts of Ravenous. So the unit of a gland is acini, which contains zymogen granules and myoepithelial cells. This is a unit of a gland, which is a serous acini, a mixed acini, and a mucous acini, depending on the type of the cell. It can be cuboidal or goblet cell, the mucous cell, and there is an intercalated duct. These have different types of secretion. The parotid has watery thin, which is rich in enzymes. The submandibular, also called a submaxillary, is a mixed with moderately viscous. The sublingual is predominantly mucus and viscid and rich in mucus. The composition and character. It's relatively large volume of around 1.2 to 1.5 liters per day per 24 hours. 20% of the secretion is from the parotid gland, 70% is from submandibular gland, 5% from sublingual gland. The pH is 7 to 8. The composition is, it has high potassium concentration with low osmolarity, and there are 2 stages of secretion, one in the acini, which is isotonic, and in the salivary duct, which is hypotonic. And there is also specialized organic materials. 99% of the saliva is water and 0.5% is solids. Of this 5.5%, 0.3% is organic and 0.2 is inorganic. The important organic constituents of saliva are salivary amylase, also called as ptyalin, lingual lipase, calcarine, IgA, lysozyme, mucin, carbonic anhydrase, blood group specific substance, nerve growth factor, lactoferrin, proline rich proteins, and can have 0.2% of inorganic, which is cations and anions. Sodium, potassium, calcium, magnesium are the cations, chloride, bicarb, phosphate, sulfate with thiocyanates are anions. The salivary composition depends on the condition of the body, the type of the stimulus, whether it is a neural stimulus, whether it is sympathetic or parasympathetic, and the rate of this secretion, and it is also under the influence of a mineralocorticoid called as aldosterone, which is a sodium retainer. The important functions of saliva are the mechanical functions. It keeps the mouth moist and helps in speech, that is articulation, facilitating movements of lips and tongue. Acts as lubricant and helps in bolus formation. Dilutes hot and irritant substance, facilitates swallowing, that is deglutition. The digestive functions are the lingual lipase, which is secreted from the Ebner's gland on dorsal surface of tongue. It is also active in the stomach and digests as much as 30% of dietary triglyceride. Ptyalin, also called salivary amylase, splits cooked starch into maltose. The optimal pH for ptyalin is 6.7. Action is inhibited in the stomach. It has an excretory function. It excretes urea, drugs, heavy metals and thiocyanates. It has protective function. IgA, which is an immunoglobulin, lysozymes, which are essential for enzyme present in the saliva, dissolves the cell wall of many bacteria and destroys them. Proline-rich proteins protects the tooth enamel and binds toxic tannins, washes down the food debris and prevents bacterial growth. It serves as a solvent for the molecules that stimulate the taste buds. It also keeps the mouth and teeth clean. It helps in the sensation of taste. It helps in water balance. Saliva keeps the mouth moist. When the body water is lost, saliva is reduced and sensation of thirst is felt. It helps in temperature regulation. It also has buffering action. Bicarbonates in saliva act as buffers. It maintains a oral pH of around 7. Saliva also helps neutralize gastric acid and relieves heartburn when gastric juice is regurgitated into the esophagus. The secretions are increased by increasing blood flow or stimulating the glandular tissue. The secretions can be under the control of nervous and hormonal. The nervous is by the autonomic nervous system, which has two limbs, the sympathetic and the parasympathetic. The sympathetic is through the cervical sympathetic chain. The parasympathetic supply is from the superior and inferior salivary nucleus. This is how the diagram is. The parasympathetic, of each controlled. The parasympathetic is from superior and inferior salivary nucleus, and each of the nerves supplying the chorda tympani supplying the auriculotemporal from the otic ganglion supplies the parotid ganglion. The superior salivary nucleus, which goes through the facial nerve and through the chorda tympani nerve, supplies the submandibular ganglion, which finally supplies the submandibular and sublingual glands. The inferior salivary nucleus through the ninth cranial nerve, the glossopharyngeal nerve, goes to the otic ganglion, where a branch of auriculotemporal nerve supplies the parotid gland. Salivary secretion is regulated either by neuronal or hormonal control. The neuronal is by parasympathetic, which is mainly by vasointestinal polypeptide, where profuse watery secretions, and it is mediated by acetylcholine. The sympathetic stimulation, which causes vasoconstriction, causes scanty viscid secretion, and its mechanism is through norepinephrine. So whenever you are tensed, the secretion of the saliva is less and mainly mediated by sympathetic secretion, so you have a dry mouth. The basal secretion increases. The reflex secretion can be either conditioned and unconditioned. Unconditioned is when the food is in the mouth and stimulus from esophagus and stomach. Conditioned is due to sight, smell, and thought of the food. The secretion decreases during sleep, fatigue, dehydration, fear, and when atropine and other cholinergic blockers are given. Salivary secretion is predominantly under neural control from superior and inferior nucleus present in the brainstem. The salivary secretion increases by parasympathetic smelling and presence of irritating food, and decreases by atropine and sympathetic. The mechanism of secretion of saliva occurs in two stages. First stage involves changes in acini, and second stage involves secretion from ducts. The acini secrete isotonic, which is sodium chloride. As the secretion passes through the ducts, under the influence of aldosterone, sodium is reabsorbed and potassium is secreted. Chloride is passively absorbed, bicarbonate is secreted by the ductal epithelium into the lumen. The applied aspects, if there is any disorders in the salivary gland, mumps is a viral infection affecting the parotid gland. It can result in swelling. Xerostomia is a condition due to reduced salivary secretion resulting in dryness of mouth. Sialorrhea is increased salivary secretion.